Hello, my name is Lara. I'm a PhD student at the University Miguel Hernández in Spain, and I'm going to talk about my first thesis chapter entitled Uncovering the Vertebrate Scavenger Gill Composition and Ecosystem Functioning in the Cerrado Biodiversity Hotspot. Scavengers include those organisms that feed on carrion from microorganisms to vertebrates. Scavenger behavior is very important for maintaining some ecosystem function by accelerating nutrient cycle, decreasing disease transmission and infection rates, structuring food webs, and stabilizing ecological communities. Among terrestrial vertebrate scavengers, we can differentiate two major functional groups. Obligate scavengers, such as vultures, are totally dependent on carrion, while facultative scavengers include other resources besides carrion on their diet. So the dependence of this resource is lower. We now know that most vertebrate predators can behave as scavengers. However, not all scavengers have the same importance within the scavenger field. Instead, there are some that play key ecological roles. Scavenger consumption patterns are influenced by several factors. Some are intrinsic to the carcass, such as carcass origin, the species to which it belongs, or its size. Other factors that influence scavenger patterns are directly related to the species included in the scavenger community, such as those with key ecological roles. Not all species are equally efficient at detecting and exploring carrion. Vultures are more specialized than any other species, consume larger amounts of carrion biomass and a higher rate than facultative scavengers, demonstrating that communities in which vultures are present have a high scavenging efficiency. It is also important to highlight the role of large carnivores, since they can either limit or facilitate the access and availability of carrion to other scavengers. There are also external factors that affect the use of carcass by scavengers, such as resource availability, seasonal changes, or habitat structure. However, the role of vegetation cover in carcass consumption patterns has been little studied, although it could depend on the sciences used by the species to locate carrion. So, the main goal of this study is to evaluate differences in scavenging patterns in the Brazilian Cerrado due to factors as carcass size, vegetation cover, and the time of carcass placement. Also, for carcass size, we wanted to evaluate if carcass size influences consumer community composition. The study was conducted in the northeastern Brazilian Cerrado. The Cerrado biome is one of the largest biodiversity hotspots on the planet, and it is composed of woodlands, savannas, grasslands, and gallery and dry forests. Livestock, mainly cattle, is present in the study area, which means that carcasses are naturally available due to both livestock and wildlife mortality. In recent decades, in Brazilian Cerrado has undergone severe transformations, and many of the habitats found in this ecoregion have been transformed into pastures and agricultural areas. But despite these threats, research on wildlife is scarce in the region and has focused mainly on protected areas and key species. In fact, the role of certain gills, such as scavengers, is still unknown. We studied carcass consumption patterns by the community of vertebrate scavengers during November 2019. We placed two types of fresh carcasses differing categorically in size: 11 large carcasses, that is, goat carcasses, and 45 small carcasses, that is, entire chickens or chicken parts. Carcasses were placed randomly and leaving a minimum distance between them. All carcasses were weighted prior to placement and fixed to the ground by placing pickets or tying them with ropes to trees or shrubs. Carcasses were monitored using automatic cameras activated by movement. We placed two cameras in front of each carcass, situated discreetly in trees or shrubs near carcasses. First, we checked the photos to identify all the consumers, the scavenger species, in each of the carcasses. A species was considered a scavenger when they were clearly detected eating carrion, so we exclude species that were recorded inadvertently. Then, to calculate the scavenging efficiency, we estimated the following variables: richness and abundance. As you can see in the slide, richness is the number of scavenging species detected consuming carrion in its carcass. That one is easy, but abundance is maximum number of different individuals of all scavenging species by identifying the highest number of the individuals appearing at the same time on an image. Sometimes counting the number of individuals is easy, as in these first two images, or in the case of these white species. But as you can see, things get complicated if we start counting individuals of the black species. We also estimate detection time, defined as time elapsed since carcass was placed until the first consumer was registered, consumption time, that's it, time passed since carcass was available until it was fully consumed, and consumption rate, that is carrion biomass consumed divided by consumption time, percentage of visited carcasses that is scavenger species, and feeding time. But we also considered external variables related to the objective of our study that could influence consumption patterns, like carcass weight, time of carcass placement, and vegetation cover. We classified the time of carcass placement in morning and afternoon. To determine the vegetation cover, we measured percentage cover of trees and shrubs within a 5-meter radius of the site where we placed the carcasses. We used polynomial analysis to determine if there were differences in the species composition of the scavenger communities of the two carcass sizes. Then we used GLMs to test whether there were differences in variables related to scavenging efficiency between goat and chicken carcasses, and to test influence of carcass weight, time of carcass placement, and vegetation cover. Moving on to the results and discussion section, this is the first time the Cerrado scavenging community is described, and our results show a highly diverse community with 19 vertebrate scavenging species, 4 species of obligate scavengers, and 50 facultative species including raptors, mammals, reptiles, and other birds. This shows that scavenging is widespread in this biome. This community is among the most diverse scavenging communities described worldwide, with 15 of the 19 of the species detected here being exclusive to the neotropical area. We find few scavenging communities with higher species richness in the literature, like the Polish temperate forest with up to 36 species, or the Californian forest with 29 species. We differentiated these 4 species of New World vultures: turkey, lesser yellow-headed American black, and cube vultures. In agreement with previous studies, our results highlight the role of other taxonomic groups being the most efficient scavengers and the main consumers, both in terms of occurrence frequency and consumption time in all types of carcasses. Also, we detected 7 species of raptors and other birds, being the soft-tailed caracara and the yellow-headed caracara the most frequent species. In our study area, facultative raptors also played an important role in terms of percentage of visited carcasses and feeding time, contrasting with other systems where carnivores are the main scavengers. Our results show that mammals and reptiles have a secondary role as scavengers in this community. Top predators such as pumas and jaguars that were detected in the area through signs and camera traps did not consume carrion, which contrasts with the important role of predators in other ecosystems such as African savannas or temperate forests. Also, current species that coexist in Brazilian Cerrado consume a great variety and quantity of fruits and insects, while in other systems where carnivores are the main scavengers, mesocarnivores like red foxes consume a greater proportion of animal prey. Permanova results show that assemblages of scavenging species consuming large and small carcasses were different. Vultures were the most efficient consumers of large carcasses, almost monopolizing them, while small carcasses were consumed mainly by facultative scavengers, a common pattern in other areas. Thus, larger carcasses could be seen as landscape of viewpoint for mesopredators and other potential prey, and therefore avoided by them. Smaller carnivores, like medium and small mammals, raptors and reptiles would avoid these reserves due to the high probability of predation on these points. Carcass size was an important factor affecting scavenging patterns. Richness of scavenger species per carcass was greater in large carcasses than in the small ones. Abundance was higher in large carcass, as you can see in these images. The left one from a large carcass and the one on the right from a small one. Consumption time and consumption rate were nearly 4 times longer in large carcasses. Moreover, no difference was found in detection time, which might be due to the high foraging efficiency of the scavenger species in the community, as happened in the African savanna. It is important to mention that the species that first detected more carcasses was the turkey vulture, followed by the southern caracara. These two species are not only guided by the sense of sight and hearing like most raptors, but also have a developed sense of smell. This clearly differentiated the New World from the Old World bird guild, giving American vultures and raptors a clear advantage when locating carrion, and therefore being very efficient, which might drive to a particular short detection time. Vegetation cover did not significantly influence the ability of scavengers to locate the carcasses, although in general detection and consumption times were higher in areas of dense vegetation or in old carcasses. Previous studies show that dense vegetation may prevent carrion localization by vertebrates, promoting carrion consumption by invertebrates, because high vegetation densities may leave insufficient space for the vulture to take off, and carcasses in open habitat are detected and consumed faster. But the factor does not seem to be relevant in our system, probably because the smell ability of the dominant scavenger species. Likewise, time of carcass placement did not influence consumption patterns. Existing research indicates that detection times should be longer in the afternoon, when mammals, which are less efficient at finding carrion than vultures and other raptors, are more active. However, the high efficiency of the vertebrate scavenger assemblage in the Cerrado reduced the importance of the time of carcass placement in this system. In conclusion, our results show a highly diverse and efficient scavenging vertebrate community in the Brazilian Cerrado biome, where we documented a total of 19 vertebrate scavenging species. We found that carcass size was the most important factor affecting scavenger kill and consumption patterns, being large carcasses dominated by vultures, while small carcasses were consumed mainly by facultative scavengers. However, we were unable to detect an effect of vegetation cover or time of carcass placement on the consumption patterns of both carcass sizes, maybe due to the high foraging and consumption efficiency of the species in this community. Finally, I would like to highlight that surroundings of our study area are highly transformed, which implies a lot of fragmentation of habitat to the detriment of the diversity and dynamics of his community. For this reason, more studies at the functional group level are still needed to understand the functioning and dynamics of communities and thus the importance of conserving them. Thank you very much for your attention. I hope you enjoyed the talk.